Okay, we are going to go through the first few slides here of chapter 24, lipid metabolism, so how our body stores and converts lipids, triacylglycerols, into energy. So my fall classes, this lecture usually falls right around Thanksgiving. So in honor of that, we have this awesome picture. And I want to remind you about lipids. We studied lipids in chapter 23, their structures, their different classes. The main one we're going to be focusing on in this chapter is a triacylglycerol. So I'm actually going to draw this out. Remember, it's a molecule of glycerol. And so we have this glycerol backbone, and then it's connected via ester bonds to the fatty acid. And since it's a triacylglycerol, tri denoting 3, there are 3 fatty acids all connected to glycerol. Okay, so remember, these fatty acids can be saturated, or they could have double bonds and be unsaturated. You can have all different kinds of glycerol stearic acid, linoleic acid, oleic acid, all different kinds. And they can vary in length. They could be 20 carbons long, 14 carbons long. But this is a triacylglycerol, and because we have these portions that I'm about to highlight in blue, all these carbon-hydrogen bonds, and this is the dominant portion of the molecule, we know that alkane and alkene type structures are not soluble in water. So therefore, let me write this in blue, this portion is not soluble in water. So the way we digest and metabolize these are going to look a little different than carbs and proteins. But the part that I'm going to highlight here in pink, all of this is soluble in water. And therefore, when we hydrolyze this, remember too, if I could draw maybe using a black line, where we hydrolyze these things with water are these bonds right here, the bond connecting the oxygen to the carbonyl carbon. And so we use water to perform hydrolysis of triacylglycerols, and the type of enzyme that will do this is a lipase. So you might hear that language in the textbook and in this chapter. This is a type of hydrolase, as it's performing a hydrolysis reaction, breaking apart glycerol, and you get the fatty acids as well, 3 of them. So just keep this in mind as we talk about the digestion and the metabolism of lipids. So also, before we jump into the details, I want to bring you back to this figure, which is figure 21.4 in the textbook, chapter 21. We have now covered all of this part of the figure in our previous chapters, and we are going to be focusing specifically on, let me change my color, this part that I'm highlighting or kind of boxing in black. All right, so chapter 24, we're seeing how this portion of the figure ties into overall view of metabolism. And so you can see the end product here is acetyl-CoA. So remind yourself, you already know the end fate here, stages 3 and 4 of acetyl-CoA once that's produced. So we're going to see how when we eat lipids, when we eat fats in our diet, what happens to them and how are those converted to acetyl-CoA if the body needs to do so. All right, so the first step of lipid metabolism is just going to be digestion. So when we consume lipids in our diet, when we consume fats, foods that are high in fats, we have to first digest them before we can metabolize them. All right, so obviously this is talking about in the mouth and the stomach. And you can see here that it says undigested fats are going to form into droplets. Okay, so why would they do that? I'm going to draw another version of what I drew in the first slide. All fats have their carboxylic acid, which is this very hydrophilic head, and then they have these hydrophobic tails. So remind yourself that this is hydrophobic. And the head, which has the carboxylic acid, is hydrophilic. So the reason they form into droplets is because this part is very water-fearing, and so they'll put these little tails, the hydrophobic tails, on the inside, and they'll surround it with the heads on the outside, kind of like what you see down here. We'll talk about this blue and yellow structure, but these would all be the very hydrophilic heads. And these can interact with the aqueous environment of the blood and the cells. So that's why they form into droplets in the mouth and in the stomach. Then we kind of have a split here, so I want to talk about this. In the small intestine, after we get some digestion, we can break apart triacylglycerols into glycerol and their fatty acids. Glycerol is soluble because, remember, I'll draw it real quick again. We have all these very water-soluble alcohol groups, so glycerol can go right into the bloodstream. There also are some soluble fatty acids if they have a very, very short hydrocarbon tail. They do have a carboxylic acid hydrophilic head, so some very small fatty acids would be soluble and can also directly enter the bloodstream. However, most fatty acids, we have to do some additional work on them, and that's going to occur in the small intestine, where our gallbladder will release bile acids, and then we have pancreatic lipases that will further cleave and work on and manipulate these triacylglycerols. So remember, real quick, let me erase my glycerol molecule here. You actually have heard about bile acids before, again, back in chapter 23, cholate or cholic acid being an example of one. Okay, we talked about how they're good emulsifying agents. All right, so I want to scroll over. The separation of the hydrophobic and hydrophilic groups allows for fats to be emulsified. So up here we have our hydrophobic side. So all these hydrophobic methyl groups. Remember these this tetracyclic ring structure is kind of the marker of a cholesterol steroid type structure, which cholate has. But on the bottom, as you notice, and I'll highlight in blue, you have all these very polar alcohol and even a carboxylic acid group, so we call this the hydrophilic side. So this bottom side will interact with the aqueous environment of the blood or the cell, but this top side, the hydrophobic side, will interact with our fatty acid and therefore allow the fatty acids to be guided through or chaperoned into the soluble aqueous environment of the blood and the cell. All right. Any other insoluble acyl glycerols, which is like a fatty acid, enter micelles. Okay, so micelles are kind of again, they kind of look down here like this blue and yellow ball-looking structure. They essentially surround fatty acids, wrap up the hydrophobic parts on the inside, and carry them through aqueous environment. They're going to be absorbed into the small intestine lining as triacylglycerols wrapped up in chylomicrons. So again, chylomicrons are kind of working just like a micelle. This right here is a picture of a chylomicron. A chylomicron is a type of lipoprotein. All right. So before I read let me read the definition first, and then we'll look at this picture a little more. So a lipoprotein, remember, a chylomicron is a type of lipoprotein. This is a lipid protein complex. So you can see, again, all these little, like this right here, these little hydrophilic heads with their squiggly yellow hydrophobic tails. These glycerophospholipids, which make up a bilayer, are kind of the surrounding part. And then on the inside, tucked on the inside, we have these triacylglycerols, we have cholesterol. These are all tucked on the inside because they're not soluble, but they're carried through soluble environments this way. So again, these are the phospholipids that are on the outside. You also do have pieces of cholesterol tucked into the membrane, just like you would into a bilayer. And we have these huge protein complexes also tucked into it's not a full bilayer here, but kind of the outer membrane portion, which is why we call this a lipid protein complex, because you got both lipids and proteins making up the whole structure. So these surround hydrophobic lipids for transport purposes. All right, so lipoproteins are the main way we transport lipids. Chylomicrons are just part of the digestive portion of this. And so they're going to carry triacylglycerols. across the lining of the small intestine into the blood. They might go to your lymph system. The delivery into the bloodstream will take them to cells and tissues, from there to the liver, so on and so forth. All right, so that's kind of the step-by-step digestion process. Chylomicrons are a big part of this. Bile acids are a big part of this. So if asked, how does the body digest triacylglycerols, be able to talk about the purposes of molecules like folate and name this. Be able to talk about how it's an emulsifying agent. Be able to talk about a lipoprotein and how chylomicrons transport triacylglycerols and why they can do that, because they have hydrophilic and hydrophobic parts. To understand lipoproteins a little bit more, because there's more than one type of lipoprotein, chylomicrons are just one type of lipoprotein. You have this picture down here. So let me read these statements first. Again, lipoproteins, these are used for lipid transport in the body because lipids are not directly soluble like carbs, okay, sugars, or like proteins in the blood and in the cells. So all fats in the body come from one of three sources. Either we eat them, they are present in our adipose fat tissue, or they are in the liver. And lipids, by the way, are less dense than proteins. So lipoproteins are classified by density according to their lipid protein ratio. So real quick, if you have a higher concentration of lipid in your lipoprotein, you'll have a lower density. If you have more protein in your lipoprotein, you'll have a higher density. So you'll see low density lipoproteins, high density lipoproteins. You may have heard of this before when talking about cholesterol levels in the body. But let's look at how this figure explains it. All right, so lipids in the diet are going to be transported by chylomicrons, which is what we discussed on the previous slide. And again, they're going to be emulsified by bile acids, transported by chylomicrons once they get into the bloodstream. Fatty acids that are stored in adipose tissue are going to be transported by this huge protein complex, serum albumin. All right, so I want to scroll out because albumin over here is really impressive. It can bind up to 10 fatty acids per protein molecule. And again, it just helps emulsify, makes the fatty acids more water soluble and transports them this way. So this is when we're pulling them out of our adipose tissue. Okay, so if we're going to if we're going to stop using glycolysis and start breaking down fatty acids for metabolism, and we'll really dive into all this step by step later. When we're pulling them out of storage, serum albumin is our complex that transports them for turning them into energy. Triacylglycerols, tags, that are synthesized in the liver, these are going to be transported by very low density lipoproteins, VLDLs, very low density lipoproteins, so mostly made up of lipids. That's why they have a very low density. And then cholesterol, so we have two different kinds for cholesterol. Cholesterol that's made in the liver, and then it's going to be transported to tissue, to cells. So the liver is sending it out to the rest of the body. These are going to be transported by low-density lipoprotein, all right? And this is really not considered a very good thing. We do have to have this to some degree happen. But when we eat diets that are really high in cholesterol, we see that our low-density lipoproteins, LDLs, are going to be heavily at work, and our tissues are going to become more filled with cholesterol because we're consuming a lot of it. So this is why this is known as your bad cholesterol, because it distributes it out to the rest of the body. This is where it can pile up in your arteries and different cells and tissues, and just that's not a good thing if we have too much of it. Cholesterol can also be taken from tissue back to the liver. So this is kind of the opposite of what we just discussed with LDLs. This is done by high-density lipoproteins, so mostly more of a protein type of lipoprotein, got more protein density to it. And this, again, is taking cholesterol from tissue. So I'm making note of that, from tissue to liver. And this is good because it's pulling it out of our arteries, it's pulling it out of our tissue and take it to the liver. So this is why, let me make a note of that. This is why this is called your good cholesterol. All right, so that kind of answers the question down here. Which kind of cholesterol is known as good? Well, that's your HDL. And which is known as bad? That's known as LDL. If you ever go get like a biometric screening done, they'll usually convey and tell you what your levels of LDL and HDL are. And it's a window. It's not a certain number that you need. It's a range of what's good and what's bad. And anything within the healthy range is considered good. So these are just all the definitions of what we said and what we talked about. And next we will cover this slide right here. So we have now finished talking about the digestion of lipids, triacylglycerols. Now what happens after we digest them? When does the body use triacylglycerols for energy? When does it store them? How does it do these things? That is what metabolism of triacylglycerols is all about. So this figure will be very handy. And while we're on this slide, we're mostly just going to be, talking about this part right up here. Okay, so I'm just going to talk about the first part of this figure, which is found in chapter 24. Dietary triacylglycerols. We know that they are converted into fatty acids and glycerol by these lipases in chylomicrons. Okay, so then we can either store them or we can convert them into energy. So when the body has a decent level of energy levels are good, we've got plenty of ATP, then we certainly don't need to take these triacylglycerols and convert them into more energy. What we're going to do is we're going to store them in adipose tissue. And cells, fat cells. So the way we do this is we're entering the figure right here. If we have fatty acids and glycerols ready to go from digestion, then instead of running it down and converting things into acetyl-CoA and ATP, instead what we're going to do is follow this blue arrow right here, this anabolic pathway, and we're going to synthesize triacylglycerols for storage. So we'll just take the fatty acids and the glycerol, put them back together into a triacylglycerol, and we'll store them in our adipose tissue. Okay, if energy levels are low, so we're low on our amounts of ATP, that's when we will take these fatty acids and we will further break them down, catabolize them into acetyl-CoA, and the pathway for this is known as beta oxidation. We will spend a lot of time after Thanksgiving talking about beta oxidation. So the final product of that is acetyl-CoA, and you know what happens to acetyl-CoA from there. Citric acid cycle, electron transport, all that good stuff. Okay, but that's just the fatty acids. The question is, what happens to glycerol? If we have the breakdown of a triacylglycerol from digestion gives us fatty acids and glycerol, we're just taking fatty acids to convert it to acetyl-CoA. We would have a leftover amount of glycerol, and we don't want to waste anything. So what happens to glycerol? We'll talk about that the next slide down. Okay, that's going to be converted into glycolysis eventually, or used in glycolysis. But real quick, adipose triacylglycerols. So it only happens when we're only going to break them down when energy is low. They're hydrolyzed to fatty acids and glycerol by lipases and fat cells, and they're transported, remember, by serum albumin to muscle and liver cells, and they're converted to acetyl-CoA. So this right here is really talking about this brown arrow, okay? Converting triacylglycerols that we have in storage in adipose tissue back into fatty acids and glycerol. So these brown and this blue arrow that I've highlighted up here are really just opposites. And serum albumin is the one that helps after the hydrolysis happens, convert it or transport it for getting it ready for beta oxidation, okay? I'm going to end this talk right here, and we will pick up with the story on glycerol, what happens to all this leftover glycerol, once we meet again in class.